Good evening, everyone. Hello, and welcome to Stonehouse in the UK for the Arboricultural Association's Wednesday webinar, sponsored by Still. My name is John Parker, CEO at the association and your host for this evening. Please do say hello in the chat, as many of you I can see already are, and let us know where you're watching from, making sure you select everyone when you do so. I always say this. Lots of you always ignore me. Select everyone, otherwise everyone won't be able to see. Tonight we will be putting Tilia in the limelight and learning all about the Wild Streets project. We're just waiting to see if Asher for the Wild Streets project is going to get here or if we're going to have a replacement. But don't panic, that's like 45 minutes away, so you can do a lot of stuff in 45 minutes, especially on Valentine's Day. Oh, yeah. Sorry, what was that? Right, yes. So first of all, before we get started, I wanted to talk about our webinar next week, which is the end of the series at the moment, unless I might add one on at the end. I'm not sure yet. I'm thinking about it, but we'll let you know anyway. It's all a bit chaotic, as you know, so we might well add an extra one on. Next week, we have two more great speakers. Levon Bigelow, Research Fellow with the Oak Ridge Institute for Sciences and Education, will be presenting his work on street tree composition and survival in Philadelphia. It's going to be really good. I've seen Levon speak before. It's fantastic. And we're also going to be joined by Karina Mafune from the University of Washington, who's going to tell us about the secret life of old growth treetops, understanding how canopy soils contribute to temperate rainforest function. That's going to be amazing as well. Really, really good, fascinating. Please do join us. You can register for both of those speakers right now, absolutely free, of course, on the website. I was going to tell you about a webinar that I'm speaking at tomorrow, actually, for the Trees and Design Action Group, but I can't remember what time it is. I'm doing something. The presentation will be ready. I'm speaking about tree management in Ukraine. There's lots of other great speakers, and Andrew's going to put a link to that in the chat as well. So you should register for that, and then you get to see me tomorrow as well, which is pretty exciting. We're definitely going to be finishing at 7:30 tonight, because immediately afterwards I will be leaving the office to entertain the ladies of the Sheepscombe Women's Institute with a talk about trees and mythology. So if we overrun, you will have the Women's Institute to answer for, and I do not recommend that you annoy them. So, use the chat for the chat, select everyone, use the Q&A button for the questions, switched on the closed captioning if that would be of help, and join me in welcoming our first speakers of the evening, all the way from the University of the West of England, proving once again that this area is the homeland of arboriculture. Our speakers, Carrie and Helene, over to you.
Thank you. I'll share my screen. Right, so I am going to talk a bit about my PhD project, and the title for this presentation is Tilia in the Limelight, Exploring the Bacterial Microbiome of Diseased Lime Trees. And let's see if I can get to the next slide. There we go. So I'm going to let my supervisor, Dr. Carrie Brady, introduce herself first. Over to you, Carrie.
Hi, everyone. I'm Carrie Brady. I'm a senior lecturer in genomics and genetics at the University of the West of England, or UE, as we are commonly known. I've been involved in acute oak decline research for more than 15 years now, and more recently, my research has expanded into bacterial diseases of other broadleaf hosts. My research interests are mainly bacterial taxonomy, bacterial interactions, and bacterial host interactions. Since moving to South Gloucestershire, I've always enjoyed visiting Westonbirt Arboretum, and I absolutely love the majestic Lime Avenue. So I was thrilled when the opportunity arose to collaborate with Westonbirt. I approached the Arboretum manager, who was Andy Bryce back in 2019, and asked if they had any diseased lime trees, which they did. So I ended up developing a master's project around the topic of Lyme diseases, basically what we thought were caused by bacteria. And Helene applied for this, and she did some absolutely brilliant research in her master's. But from there, it took a good couple of years to gain the funding to design a PhD project around this. But finally, in 2022, all the stars aligned as we were able to gain funds for the project with a very grateful contribution from the Friends of Westonbirt Arboretum, as well as UE, and fortunately, Helene was still able and willing to take on the project. So now I'm going to hand over to Helene so she can tell you all about the research she's been doing so far since she started January 2023. Helene?
Thank you for that, Carrie. So my name is Helene Kile. I'm a second year PhD student, and I grew up on the Norwegian countryside, kind of surrounded by pine trees, which is where my interest in the natural world started. And before starting my studies in the UK, I worked as a veterinary nurse for several years, which I very much enjoyed. However, I was aspiring for a more ambitious career. So in 2017, I moved to the UK and I started my bachelor's in biology. And during these years, I especially found plant biology and plant pathology interesting, which then led me to undertake a master's in research that Carrie mentioned. That later led to this PhD project. First, I wanted to start with an introduction to lime trees in the UK and Europe. So when I refer to lime, I refer to the broadleaf lime or linden or Tilia tree, so not the lime fruit producing tree. Only 3 species are native to the UK, and that includes the large-leaved lime, Tilia platyphyllos, the small-leaved lime, Tilia cordata, and their hybrid, Tilia europaea. These are quite large trees that can reach 30 to 40 meters in height, with a trunk diameter of 1 to 1.5 meters when they're about 300 years old and growing in natural habitat, such as a woodland. And the species are found naturally throughout England and into the east of Wales, but they only occur as planted trees in Scotland and elsewhere. Lime trees have a poor ability to colonise, so therefore they're often rare or absent in new forests, and they are more frequently found in old woodlands. And because of that, they're also considered ancient woodland indicators, which means that their presence can be used to recognise these biologically important woodlands. In urban areas, such as parks and avenues, Tilia cordata and Tilia europaea are among the most frequently planted tree species. So you can kind of divide the population of lime trees into the native or naturally occurring population and the planted population. So if you have a look at the images, the blue image shows the native distribution of Tilia cordata, while the red image shows the distribution of Tilia platyphyllos. And as you can see, Tilia cordata is more frequently distributed. So a bit about the importance. So the importance of the species can be highlighted through the range of ecosystem services that they provide, both to UK woodlands and also urban forests. So lime trees have been reported to contain high concentrations of nutrients in their leaves. They decompose rapidly, and that positively influences the soil. In urban areas, they provide several benefits, including improved air quality as they remove pollution, and they also avoid stormwater runoff. They support the pollinating community, and old hollow lime trees, both existing in woodlands and parks, they provide valuable habitats for insects and beetles, including red-listed ones. In terms of uses, the tannin in the flower of several lime species, including Tilia cordata and Tilia platyphyllos, is used to make tea and herbal extracts, particularly in Eastern Europe, where it is actually used for the treatment of colds and coughs. And the wood of lime trees is very flexible and strong, and that's suitable for veneer, musical instruments, and hand carving, and is traditionally used throughout Eastern Europe. But despite Tilia cordata actually being one of the most widely distributed tree species in Europe, its economic value in the West is low. And unfortunately, over the last 2,000 years, the distribution of lime trees in Europe and European woodlands has declined. And that is mainly due to the exploitation of beech, but also from farming expansion and low seed fertility. So the naturally occurring population is now considered highly fragmented, which leaves them vulnerable. And the main reason for the decline is the low economic interest for lime trees, which then has led to the replacement by higher valued timber species. However, neither of the native species are regarded as threatened in the UK, despite this declining population. And it's also really important to mention the threat of climate change, as it's predicted to negatively impact forest trees and to accelerate the spread and damage of pests and pathogens. So lime trees have a reputation of being resilient towards disease. But despite that, reports of Phytophthora infections have increased over the past decade. And the genus Phytophthora consists of a diverse range of pathogenic oomycetes, which is a fungal-like organism that infects a variety of UK native trees. And the pathogen commonly spreads through soil and watery soil, as it produces zoospores that have tail-like structures, which enables them to move and swim through water bodies, and then infect neighboring trees as they penetrate the roots. And infections caused by Phytophthora species has led to serious outbreaks worldwide, and the route of infection for several of the UK outbreaks can be traced to imported planting material. Common symptoms of infection include dieback of the canopy, needle and shoot blights, necrotic stem and branch cankers, and the disease can cause mortality. And importantly, there are no effective treatment options available. In lime trees, cankers on the trunk of the tree are a common symptom of infection. And as you can see in those pictures, the canker stands out as it's quite dark in color compared to the grayish bark of lime trees. Sometimes the cankers are dry, while other times they're moist, and occasionally they're wet or even slimy with exudate seeping down the trunk of the tree. And that is also referred to as bleeds or bleeding cankers. So overall, the severe economic and ecological implication caused by Phytophthora, combined with a lack of effective treatment option, really highlights the importance of continued research in the field. So moving on from one tree species to another, I'm just briefly going to talk about acute oak decline, as it does link to my project. So this disease affects mature oaks over 50 years old and can cause death within 4 to 5 years after they first show symptoms. And the symptoms are commonly observed are cankers and necrotic lesions on the trunk of the tree. And 2 bacterial species, specifically Brunneria goodwinii and Gibsiella quercenicans, are involved in the diseased complex, causing tissue necrosis. And these bacterial species have also been isolated from the bleeding cankers of lime. So as I mentioned in the introduction, or as Carrie mostly mentioned in the introduction, my work on lime trees started with my master's project. And for that project, I isolated and described a new bacterial species named Tilia. And it was isolated from Phytophthora-induced cankers in lime. And that is interesting because this bacteria belong to the Brunneria genus, which consists of several known tree pathogenic species. So additionally to Brunneria tiliae, two other bacterial species, Brunneria goodwinii and Gibsiella curicinicon, so the pathogens associated with acute oak decline, were also frequently isolated. And based on these findings, it was hypothesized that they may have a pathogenic potential within the canker microbiome of lime trees. And that led to several questions being raised regarding bacteria. And my PhD project, it builds upon these previous findings. And yeah, the PhD is funded by Friends of Western Birch Arboretum, which is a charity, and also UE. So I'm going to now tell you a bit about what I'm trying to achieve. So the main aim of the project is to locate, survey, and sample lime trees suffering from Phytophthora-induced cankers to do a microbiome study. And the results from healthy and symptomatic trees will be compared to determine differences and also to pinpoint potential pathogenic bacteria. So those selected potential pathogenic microorganisms that are found will be further investigated through pathogenicity tests to determine whether they can cause disease in lime trees. And importantly, we plan to include all of the native species of lime for the pathogenicity tests to find out whether some may be more resilient than others. And results from this project could give us some new insight regarding the role of these potential pathogenic bacteria within that canker microbiome, and therefore the threat that they pose. It would also give us increased knowledge about Phytophthora infection in lime trees, which I think is an understudied research area. And the resilience study could influence and benefit future planting strategies for lime. The focus for the first year of the project was to locate sites with symptomatic trees to visit. And for this, we explored personal networks, we researched lime tree sites in the UK and engaged with the public. And that was through social media and information leaflets. And that proved to be very successful. And we had plenty of help from professionals within the tree sector, as well as volunteers who reached out and even sampled trees for us and delivered the swabs by mail. And this massively contributed to the data collection. So once we had located sites, we tried to visit as many as possible in order to survey and sample the trees. And for the survey, both the disease symptoms and the general health of the trees were described. And for the sampling, I would use a cotton swab to swab the moist exudate from the cankers. That's where the bacteria are more likely to be present. And yeah, as the image shows, swabbing away at a canker. And while I was still in the field, I also made sure to perform a Phytophthora rapid test And I would use a bit of symptomatic tissue for this test. And that's a lateral flow test, very similar to a COVID test. And as you can see on the bottom image, it shows a positive result, positive Phytophthora result with 2 clear lines. So here I've included some images taken during some of the sampling trips. So the top image to the left, it shows a rather dry canker, and there's also a crack in the outer bark layer. Upon removal of the outer bark, the lesion on that second image was discovered. And the 2 images below, they show a tree with a very large canker. I think it was more than 1 meter, probably in length. And similarly here, we removed a bark panel to try and reveal what was going on underneath. And in contrast to the image above, it didn't expose a necrotic lesion. However, the tissue was abnormal with high amounts of tannins present, which explains the orange color. And it's likely that Phytophthora is breaking down the tissue, and that's what's going on. The images on the right show a selection of cankers that we encountered. Some were dry, others were moist, as you can probably see in the images. And that is what we would expect to see when inspecting lime trees suffering from Phytophthora infection. So after the field trips, the samples were brought straight to the lab for processing. And I just thought I'd talk you through that procedure. So after swabbing, so in the lab, I would smear the swabs onto an agar plate to promote bacterial growth. So that agar plate contains nutrients that will enable the bacteria to grow quite rapidly. And I would then incubate the plates for 24 to 48 hours until the bacteria had grown sufficiently. My agar plates would then have lots of mixed colonies of bacteria growing. So the next step was to pick all the unique colonies and then restreak them to get pure colonies. So I would restreak them onto fresh agar and then incubate them again. Once ready and pure, I would extract the DNA, followed by PCR to amplify a specific region in the genome that is useful to determine the identity of bacteria. Next, I would send the samples off for nucleotide sequencing of this specific amplified region. And based on the results from searching in database, I could then in most cases determine their identity. If the bacterial identity was unclear, for example, if we had a new species on our hands, I would draw a phylogenetic tree to determine the correct identity. So a total of 60 sites were included, and the sites were either visited, inspected and swabbed in person, or the swabs were kindly sent to the mail by volunteers and professionals. And the northernmost area that was included was East Lothian in Scotland, while the southernmost was Plymouth in Devon. And to the east, we had Santon Downham in Suffolk, and to the west, Attingham Park in Shrewsbury. So quite a nice, good spread of sites. And several local sites were also inspected, including Western Birch Arboretum. The results from the Lyme surveys reveal that the symptoms and the severity of the disease varied across sites. And also among trees within each site. So in general, the nature of the cankers was rather uniform. with the discoloration of the bark appearing as brown to dark brown and occasionally nearly black. But some cankers were wet, other were dry, and bleeding episodes also seem to fluctuate, and it seems to correlate with rainfall. So with more rainfall, the cankers would seem more moist. The size, location, and the number of cankers present also varied. Some trees had multiple cankers, while others only had a single small one. And similarly, the general tree health and canopy cover also varied distinctly among trees. So, yeah, the general health didn't always correlate with the severity of the cankers. So to demonstrate that, here is Tilia europaea that we visited in Attingham Park in Shrewsbury. And as you can see, the canopy is very nice and full with no visible sign of dieback. However, the trunk did show quite a lot of bleeding cankers, and there were more cankers on the other side of the tree as well. And then this tree, located in Matlock in Derbyshire, this is also a Tilia europaea. It displayed severe dieback of the canopy. However, we only noticed a few quite small cankers on the trunk. So regarding the results from the microbiome studies, Phytophthora was identified at every site that we tested using the rapid tests. And in terms of bacterial isolations made from the swabs, frequently isolated bacteria of interest included Brennera tiliae and also the pathogens associated with acute oak decline. On the table to the right, that shows the number of isolations made across the sites, and Brennera tiliae was isolated from 9 out of 21 sites, Brennera goodwinii from 12 out of 21 sites, and the same number was found for Gibbsiella carcinicans. So what do the results mean? Phytophthora is likely the main pathogen causing the cankers in limes. However, the species is unknown. Several potential pathogens were frequently isolated from the canker microbiome, and that could indicate that they contribute to disease progression. And I hypothesize that these bacteria are opportunists who take advantage of the situation. So let's say normally these bacteria reside within the host in small numbers as endophytes. But when Phytophthora infects the host, it breaks down the plant tissue, which leads to the release of nutrients. And then this sudden abundance of nutrients could open up an opportunity for these bacteria to proliferate and then also potentially contribute to the breakdown of tissue and therefore disease progression. However, we don't know if these bacteria already exist as endophytes inside the host. It could also be that they are secondary invaders, taking advantage of the reduced host immunity caused by the Phytophthora infection. That kind of remains to be determined. So the way forward is to firstly perform metagenomic analysis of 3 to 4 selected sites with symptomatic trees and 1 site of healthy trees. And so by removing the outer bark layer, I want to sample the inner bark of the lesions by taking shavings of this symptomatic tissue. And the samples will then be processed in the lab, and the DNA will be extracted and sequenced. And that will provide a complete overview of the microbial composition. And the advantage of this method compared to analysing single swabs is that we get much more data. And in addition to the bacteria present, we will also learn about the fungal and mycete community. It's also unbiased compared to the swab analysis method. And that is because it will reveal information about the non-culturable microorganisms as well. So the ones that won't grow in the lab, basically. And I would then compare the data from the symptomatic trees to the healthy ones to then pinpoint differences and potential pathogens. Next, pathogenicity tests will be undertaken, and they will include bacterial strains selected based on the metagenomic analysis, and they will also include Phytophthora. So for this, line saplings will be inoculated with the bacteria and then monitored over time to see whether necrotic lesions develop. And at the end of the experiment, the lesion size will be measured and statistical analysis performed to determine the significance. So, in other words, whether the bacteria can cause disease. So overall, this will provide clues regarding the role of selected potential pathogenic bacteria in the canker microbiome and the potential threat posed to lime trees. Thank you so much for listening and for inviting me to chat about my project.
Brilliant. Thank you very much indeed. That was great. Good stuff. I can't see Helena or Carrie at the moment. I'm just throwing it out there. I'm assuming you're still there, but I'm going to... I'm
trying to figure out how to put my camera back.
No worries. There you are. Okay, so just so you all know out there in webinar land, unfortunately, we have now lost our second presentation. Boo. Because Asha was unable to attend, and she tried to get a colleague to cover, but couldn't cover. So we have a bit of time for questions, and then we'll see how we go. Maybe we'll freestyle another half an hour a la Harry Studholme. So we've got a few questions in here. We'll work through some of the most interesting ones, but I don't know if you can see the questions there as well. If you see anything you think is particularly interesting that I haven't mentioned, please do let me know. Trevor's asked about the age of trees that are being tested. Are all varieties of each type tested?
So regarding the age, the majority of the trees are mature lime trees. So I would guess they're between 50, so semi-mature, mature, 50 to 200 years old, planted trees. And the majority of the trees that we've included so far are Tilia, europaea, because they are more frequently planted, especially in urban areas. And we tend to see that they might be more prone to disease because we do seem to find them more frequently than naturally growing natively distributed trees.
Don't forget though that the whole project did start because the diseased lime tree at Westonbirt was a different Tilia, wasn't it? It was a Tilia mulci.
It was, yeah.
And we do know that a couple of other quite unusual Tilia, they're hybrids of species, have been found with bleeds at Westonbirt as well, but they're quite small. Was the Tilia distilia ringing a bell?
Tilia japonica and
yeah, was it I can't say it.
They're not UK native ones. They're quite rare, but it's an arboretum, so they do have a very extensive collection of tree species.
And this is a question from me. Be honest, were you a tiny bit disappointed when you got to Western Burt to see the 2,000-year-old lime tree and realised it was a coppice? Because I went up there going, this is going to be a monster, and it wasn't.
Yeah, I a little bit. I must admit, I didn't think it was actually that one, so I kept looking, but it was, yeah.
Yeah, I mean, Western Burt's amazing. Everyone should go. You should all go and look at it. And there's wonderful trees. But yeah, I was like, oh, surely not. Anyway, right. Excuse me. Oh, people are getting very technical. Paul, you always get very technical, but we're going to go with it. Paul asks, does potassium phosphate PO3 stem injection just improve cellular defense against bacterial and fungal pathogens, or does it actively affect the infection also?
Helena, should I take this one?
Yeah.
Short answer, I don't know. But I do know that bacteria can solubilize potassium, and in some species they can solubilize it and then they can use it to promote the growth of the plant as well. So they are, say, more plant growth promoting bacteria than plant pathogens. But I would have no idea about if an injection into a tree would harm the tree more at the site, cause, I don't know, additional weeping, or I doubt that something like that could tip the odds in the favor of the tree and cause the bacteria to start promoting its growth. But yes, short answer, I'm sorry, I don't know.
Thank you. There's some more technical questions from Paul there, but we'll come to them in a little bit. Martin has got a question about how worried we should be, basically. How concerned should we be about the potential for mass demise of our lime tree stock in the UK?
At the moment, this research is very much in its infancy, so it's very difficult to say something about that. But I'm hoping that this work will give us maybe some indications of the future threat posed to lime trees, and especially depending on those pathogenicity trials. I hope that we can find out more. We know that Phytophthora is already it's a big problem in the UK, and a lot of lime trees are affected. But yeah, the research isn't that great, I think, on lime trees, perhaps because the majority of trees are planted and they grow quite easily and rapidly in urban environments, and if they get diseased, they get chopped down and replanted. So yeah.
Brilliant. Yeah, it's worrying because we always we know about so many pests and disease that affects so many of our trees, and lime has always kind of felt like one that has kind of been okay. But maybe not.
But I think what we have noticed is we haven't seen trees that are recovering from the cankers, whereas with other tree diseases, sometimes they can heal themselves, they can callus over, but we haven't found many sites that we've visited where we've seen trees improving with health. Once they have the cankers, they seem to stay or get progressively worse. I think there's a site that Helene found in Minchinhampton that she's periodically been back to, and a very old tree there. It's just getting more and more cankers, and we think it have to eventually be felled.
Thank you very much. There's lots of questions. What shall I go for next? Oh, Daniel's asked if you're still looking for any study sites, and if so, how can people get in touch? Is there any sort of database they can help with?
Sorry, what does study site?
Study sites for, I guess.
Study sites. Sorry. Okay. Yeah. Okay. I'm with you now. Yeah, we're always interested in new sites. Every site is really valuable. I'm wrapping up now with the swab analysis part of the project. I think, well, I need to move on and prioritize the metagenomic work. But we're just happy to have like a nice overview of symptomatic sites, and if it stands out, for example, we might want to include it. So yeah, definitely.
What I can do is we have an information leaflet that we made at the very beginning part of the project, which we were distributing through our contacts at Forestry England, and that's where we received the majority of our samples from, was this leaflet being picked up and passed around to local tree health officers. I'll put it in the chat. It's got our details on, so people have just been emailing us. Some people already have swabs. They would just swab the tree and put it in the post. But what's important for us is location. So we use what 3 words? So a what 3 words location would be great if you have a site, and then photos if possible, and just a bit more information, because it just gives us a really well-rounded picture when we're plotting them out. So yeah, I'll put that in the chat now.
Brilliant, thank you. And I was going to mention as well, it was only through Western Burt and the Friends of Western Burt Arboretum that I first saw your research as well, reading in the magazine there. So that's when I first made contact. So yeah, it's well done, Western Burt. I'm biased because I was a trustee of the Friends of Western Burt Arboretum, but they are. I wasn't a very good one, but I was there. Okay, Iona has asked if there are similarities in the genome of bacteria that affect Tilia and Quercus.
Should I take that one, Helena?
Yes, please.
Okay. So I've actually have just had a small project on this topic. So I had a postdoc who sequenced a whole lot of Brunneria goodwinii genomes. So Brunneria goodwinii is the species that's mostly responsible for the necrotic lesions in AOD, and also the species that we sometimes, not all the time, find in bleeding cankers of Tilia. So we've sequenced about 60 genomes. Most of them are from oak. We have 1 or 2 from lime, and the analysis is being done on that basically as we speak. In terms of, I would say, similarities, it's one species. They will be very similar, but we are finding small differences in things like the virulence genes that the bacteria have, the different strains of Brunneria goodwinii, and yeah, possibly some differences in the extra DNA that the bacteria carry in terms of plasmids and things. But it's all still ongoing. But yes, we are looking at that.
Thank you. Alistair's asked if you observed any correlation between the severity of the canker appearance and which combination of bacteria were present. Do any particular combinations manifest themselves more obviously?
Yeah, that's a very good question, and I think that's something I'm going to look into when I do some more data analysis. When I kind of wrap up this or the swab analysis, I will start having a look at and then compare results. So at the moment, I don't know, but hopefully I would be able to answer that in the future.
Brilliant. So you have to come back for another webinar when you've done some more research. Good question. Right. Chris has asked if you're testing wild trees as well as planted specimens.
I haven't been able to locate any wild trees with cankers yet. It's something I would be very interested in, but we haven't at this moment found or heard about any sites where natively distributed trees are suffering from cankers.
And I mean, is that quite interesting finding in itself, the fact that the naturally occurring ones you haven't found any cankers on, but you have on planted?
Yeah, I also think that I don't know, perhaps they're not that frequently distributed in the wild compared to the planted population, so that could be a reason why as well. And especially like in urban environments, people are bound to notice bleeds, and they might be surveyed more frequently as well, which might impact that. So they might exist in the wild as well. We haven't located any.
Brilliant.
Yes, if anybody does find any, please do let us know.
Yes.
I was just about to say we got 450 odd people who are watching at the moment who will all be out there rooting around their lime trees to have a look. So you're going to be inundated with data. So Emma's asked, when specifying limes to plant in the UK, what's the advice? Is there some to particular varieties to go for or to avoid, or have you got any advice generally for those looking to plant limes?
No, I don't have any like direct advice. We don't know whether some might be more resilient than others. The most commonly planted ones is the hybrid Tilia europaea and the small leaved lime Tilia cordata. Tilia platyphyllos, not so much. But yeah, at the moment, I don't know whether there's a difference in resilience between those two. But genetically, they would be quite similar since one is a hybrid. So yeah, not sure.
Thank you. Padraig has asked if climate change is likely to speed up the progression of this disease.
Yeah, we think so. And as I mentioned in my presentation, the occurrence of Phytophthora in lime trees has increased during the past decade, and we think that's due to climate change and also increased like imported plant materials. But in terms of climate change, it's likely to impact the natural immunity of the host and these internal stresses that occurs during drought episodes, for example, does cause the host to become more vulnerable to pathogen attacks. So yeah, definitely believe it's involved.
And that slightly ties into another question from Paul, which you may just have answered. But Paul said he's seen a rise of Phytophthora in lime trees in the north of England and Scottish borders in the last decade. He's saying he's seeing it as a stressing pathogen, which doesn't seem to kill or decay the trees significantly unless the tree's been stressed already by other factors. Is that what you've noticed? It seems to be from what you just said about the climate issue.
Yeah, I think so. Yeah.
I think it's something we'll also factor into Helene's pathogenicity tests as well, is that we will have a set of trees that we are going to stress, or saplings that we are going to stress. Maybe we will withhold watering, try and simulate drought conditions, or ring barking of the sapling. I don't know if that would stress them as well, just to see if that is a difference between lesions appearing or not appearing, is if we stress the tree and then see if disease develops. I'm just frantically reading in the chat as well to see if there's any other questions that are coming up.
Don't indulge them. We can, but yeah, don't feed the monkey. The clear instructions here, folks. Questions in the Q&A panel, otherwise we can't keep on top of it. Sometimes I do let them off if they put in a particularly good one. Benjamin's asked if we know how far it's spread across Europe.
Phytophthora.
In limes, I guess.
The limes, yeah.
Sorry,
if there's any, we don't have many reports actually. If we have any reports in Europe, there's not much. There's been reported. There's not much in the literature on this. So we basically just have to focus right now on what's happening in the UK and see if that what we learn could possibly be applied to Tilia in Europe. Yeah.
Thank you. And I should have said, of course, because someone will say it in the chat in a minute. Of course, the UK is in Europe. I think.
I did see that in the chat.
I haven't seen it yet, but I know someone will say it. But of course, we are in Europe, so that's good news. Right. What have we got here? Ward has asked if any treatment's been tried with heat, just like they have trials with heat treatment against bleeding canker with chestnuts.
We haven't tried that at the moment, but it's something we would like to include in when we get to the stage of doing the pathogenicity test to expose the inoculated saplings to varying temperature and also levels of moisture, so drought, and to see the effect of that, whether it potentially increases the pathogenicity of both Phytophthora and also the bacteria.
Brilliant. Thank you very much. I'm just if either of you have seen any questions in the Q&A or the chat that you want to address and I haven't, then please go for it.
I would just like to answer Bill's question when he asked, could we please repeat the word oomycete? Because he's been wondering how to pronounce it. So, oomycete.
Yeah, I'm probably mispronouncing it. I say oh my seat, but oh my seat.
Oh, my seat.
Yes, I need to practice that.
It's a tricky one.
Yeah.
I've definitely been pronouncing that wrong then.
I could just be, tomatoes, tomatoes.
Okay. Ladislav's asked about, is there any research on the gut microbiome of bark beetles where these bacteria were present?
So yes, I can take that one. I actually am involved in a collaboration with Forest Research at Alice Holt in Surrey, and they've been trying to look at the gut microbiome of Eucalyptus beetles for a while. It's not always so easy to do, just having access to the beetles, but they have managed to rear them in captivity, and we've just finished an experiment where the beetles have been fed on lab-grown leaves, say leaves from a forest, and leaves that have been dipped in the bacteria to see if the bacteria can survive in the gut of the beetle. And that research is ongoing, and we are looking for further funding for that. But yeah, it is being done.
Thank you. I'm going to ask another question from Paul, and I'm going to practice the word I just learned to say. Have we worked out what oomycetes are native to Britain and what is imported? Is it assumed all pathogenic strains are imported?
Oh,
yeah, you go for it, Carrie.
Yeah. Again, not too sure on that. I think there was a question as well, just to link in with that, where someone was saying, is the Phytophthora spread by the rhododendron? Not the species that we think is in the lime. It's really hard to isolate Phytophthora from the wood or from anything, and to characterize it is even trickier. Yes, so in terms of that, I'm not too sure. And the ones that are regularly, I say, flagged up by DEFRA, I'm assuming that some of those do come in from imported material. But Phytophthora is everywhere. It's in the soil. I think it's just really taking advantage of stressed trees or plants. But yeah, it's sorry, I was reading about the my seats again in the chat. Yeah, it's just really hard to say.
Thank you very much. So we probably have time for one more question before we move on to the final bit. I don't know if you've seen anything you particularly want to address, or I can just pick one at random, basically.
I don't know if you've been looking, Helena.
Yeah, no, you can pick a random one.
Pick one at random. Okay, well, I'll go for Oh, sorry, Carrie, have you got one you wanted to?
I was to say there is a question from Anonymous who asks, how can you determine that different organisms are disease-causing rather than just growing on dysfunctional wooden trees under some other stress? That is also kind of really tricky, say, to do/answer. But in the pathogenicity trials, Helena will be looking at different combinations of things. So she will have sets of saplings that we will just wound but not inoculate with anything. We will have saplings that we wound, inoculate with just the Brunneria tilia. We will have sets of trees with combinations of AOD bacteria and Brunneria tilia. We will hopefully be able to isolate the Phytophthora and include that in the combination, and then also stress the trees in different treatments. So we'll work out an experimental plan with lots of different combinations of things to try and exclude that option where that it's just the bacteria is just there and growing on something that is stressed, to really determine which one is pathogenic.
Thank you so much. Absolutely fascinating stuff. Okay, well, I know we're finishing early. There'll be people going, boo, but hey, we've all got things to do, I'm sure. So we will wind up with a final question to both of you. I'm not sure who wants to go first, but Helena and Carrie, what is your favourite tree-related book?
Yeah, I guess I can go first. Well, I first encountered this book when I started researching lime trees during my master's, and it's a book by Donald Piggott, and it's called Lime Trees and Basswoods, and it's a biological monograph of the genus Tilia, and it focuses on taxonomy. And it was very valuable for me because I was trying to learn more about lime trees and also being able to differentiate them, which can be quite tricky, especially the hybrid. And this book is very well written, it's very detailed, and it has some wonderful drawings. So yeah, it was very, very useful for me. So I recommend if you want to learn more about lime trees and the different species.
Wonderful, and that is we've not had that book before, I can tell you, so that's very good. Fantastic. Carrie, what's yours?
So mine is about oak. It's called The Glorious Life of Oak, and I always forget the author, so I had to get the book out and have it next to me so I could say it's by John Lewis Stempel. And oak and oak disease kind of started off my own research career, so it's very special to me from that point of view. And it's beautifully written. I love all the mythology around oak and the historic aspects of it as well, and he captures that really well in his book. Hold the books up to the cameras. I don't know if you'll be able to see that. It's like
with a blurry background, it sometimes just sort of blurs, but there you go.
Sorry about that. Yes, that's my favorite book. There was a question in the chat how people can get hold of us if they would like to. So I don't know if you want to contact us through John. He can forward messages on to us. Otherwise, you can basically just search Carrie Brady, UWE, and my details should pop up.
Brilliant. And excuse me, like we always say to everyone, folks, we'll send the chat and we'll send the questions to our speakers and they can have a look through them. And then if you've got anything additional, please do send it on to me or to the email you've sort of registered with here. So brilliant. Well, hope to see you all next week for what may or may not be our last webinar of the series. I'm not entirely sure. We'll see how things go. But Helena and Carrie, thank you both so much. That's absolutely fascinating research. Well, people always ask. I think we had just under 500 people watching, so that's my sort of estimate. So it's around there, which is a wonderful audience. So thank you both so much. Apologies for the short finish, but you can go and have fun doing your thing now, folks. Hope to see you next week. Thank you to Steel, of course, our sponsors, and we look forward to seeing you very, very soon. Take care. Bye-bye.